Cloning in animals and transfection methods. What is cloning? Formation of one or more genetically identical animals from a single parent animal is called animal cloning. Your monozygotic twins are a clone of each other. They develop from one zygote by splitting of the early embryo. Zygote is unicellular whereas your embryo is multicellular. Zygote, we say it is just after the fertilization of egg. Later, your zygote develops into an embryo. At that particular stage, zygote undergoes splitting in this case. Let us know about the history and some developments in cloning of animals. The first successful experiment in animal cloning were performed by Garden in 1962. He was successful in his experiments on toad. They are similar to frog. Next, Wilmot and co-workers in 1997 from Scotland, they produced the world's first cloned mammal, a sheep called Dolly. You must be heard of the name Dolly. It is a first cloned mammal. Then scientists from USA have recently cloned an endangered species, that is Asian gur. Japan scientists have cloned cattle in a different way, that is 8 identical calves from one fertilized cell of their mother. They have succeeded in growing as many as 8 identical calves from one fertilized cell of the mother. Let us know what is there in Gordon's experiment. He takes the epithelial cells of a tadpole. Tadpole is nothing but it is a young toad. Okay, adult will call them as toad and the young ones are called tadpoles, but they are the same organisms. Okay, they are similar to frog. The limbs of the frog will be taller when compared to the toad, and the skin of toad will be hard, and the frog's skin will be slimy to touch. So the intestinal epithelial cells of a tadpole were taken. Then the nucleus from the epithelial cells are taken out. Okay, you can see the steps in the picture clearly. Follow by seeing the picture. Okay, epithelial cells of tadpole are taken. From those cells, the nucleus is separated. On the other side, unfertilized egg is taken from another toad. So that unfertilized egg is taken from the other toad. There again it is taken without nucleus, that is it is denucleated, and the other functional organelles of the cells are retained, and that is taken. So here the nucleus from the epithelial cells of the tadpole and the denucleated cell are allowed to fuse together. Okay, then after fusing they develops into blastula stage and finally to tadpole and the fully grown adult, that is toad. So here the egg underwent normal development and produced a toad. The resultant will be the clone of the Todd which donated its nucleus. Here which one donated nucleus? The tadpole, from which we have taken the epithelial cells, no? From those cells we have taken the nucleus. Here the resulted Todd, what we here you can see the Todd developed, it will be the clone of that particular tadpole. So this is about Gardner's experiment. The world's first cloned mammal was Dolly. You can see that sheep in the picture. In 1997, Wilmot and co-workers produced the world's first cloned mammal, that is a sheep named Dolly, on February 13th. How the sheep Dolly cloned. It was a major development in animal cloning actually. Subsequently, several cloning experiments have been performed later on. How it is formed? Here they took the cells from the udder of a 6-year-old sheep. Then they managed to store these udder cells in nutrient deprived culture in order to deactivate the genes present in the udder cells. Then one udder cell complete with its nucleus was selected, as this nucleus carries the mother's genetic information. Then on the other side, unfertilized egg of another adult sheep was taken and the egg was denucleated. Non-dividing nucleus of an udder cell was taken out and inserted in the denucleated egg. Okay. Then now they get fused, udder cell nucleus with an empty egg cell by electrical stimulation. We have to apply electrical stimulation in order to fuse the cells. Then this egg cell will be having mother's nucleus. In nutrient medium, the egg then began to undergo cleavage. The young embryo was then transferred or implanted into the uterus of a different sheep, and that sheep will be called as surrogate mother. The surrogate mother gave birth to normal healthy lamb, that is Dolly. It is genetically identical to the mother sheep. So let us see the flow chart, then you can understand it better way. So in the picture you can see sheep 1 and sheep 2. So from sheep 1 the udder cells are taken, and from sheep 2 the mature ovum or egg is taken. The nucleus is removed from the udder cells, and in that egg cell empty ovum is taken. So nucleus will be from the sheep 1. Then they are cultured together. Then the mild electric shock will be given and electric stimulation will be given in order to fuse the nucleus and the empty ovum. So they gets fused. So here nucleus from sheep 1 fuses with the empty egg from sheep 2 and starts to divide to form an embryo. Then that will be inserted into the uterus of the surrogate mother, that is sheep 3, The cloned embryo is implanted into the uterus of sheep 3. Sheep 3 will be the surrogate mother. Then it will give birth to a lamb. Lamb born will be the clone of sheep 1 now, because it carries the nucleus from the adder cells of sheep 1. So this is how the dolly is formed. Okay. So next will be the cloning of cattle. So scientists from Japan have cloned cattle in a different way. They have succeeded in growing as many as 8 identical calves from one fertilized cell of their mother. When the mother cow has mated with the bull, she has fertilized egg, that is zygote, in her womb. Okay, this cell divides into 2 and then into 4 and then in 8 in number. Okay, in the picture you can see the cow and the bull, they are mated. The egg is fertilized with the sperm cell from a bull. Then the original embryo from the womb of cow is removed. Okay, the embryonic cells are then separated using an enzyme. Okay, 8 cell stage once it gets developed, then or 4 cell stage once it gets developed, at that time it is removed from the womb of a cow. Then with the help of enzymes, these embryonic cells are separated. Okay, then each isolated cell is kept in a nutrient medium, and later those will be implanted in the womb of a different host mother or cow. So in the picture you can see the original embryo are allowed to develop into original cells are allowed to develop into embryos. Then they will be inserted or implanted in the womb of the different cows. The host mother's womb must accept the cell and should make it grow. Okay. Each cell grows into a normal, healthy baby calf. Then the host mother gives birth to calves. This is about cloning of cattle. Clone of Asian gur. Scientists of USA have recently cloned an endangered species, that is Asian gur. Its scientific name is Bos garis. It is an humped horned mammal. It was cloned from a single skin cell taken from a dead Asian gur. As I told you, it is an endangered species. Single skin cell is taken out from a dead Asian gur. Then the skin cell was fused with a cow's egg. Okay, it is fused with the cow's egg whose gene was removed. This step we have learned in the last example. The fused cell was transferred to the uterus of another cow. Okay, the skin cells from dead Asian gur, and then it is fused with the egg from the donor cow. From the egg, the gene was removed. Now the fused cell was transferred to the uterus of another cow or surrogate cow. Then the resultant, as a resultant, we see the gur calf was born. So it as it contains the nucleus from the skin cell taken from the dead Asian girl, the calf shows up that particular character. So the girl calf was born. Even though the mother is cow, it will show up the characters of the Asian girl because it contains the nucleus, genetic material from that particular organism. So Asian girl is the first endangered species to be cloned and also the first cloned animal to gestate in the womb of another species. So let us know about some transfection methods. In animals, transfection specifies the introduction of a DNA segment either naked or integrate the DNA segment integrated into a vector then into an animal cell. So the gene introduced by transfection is called a transgene. So what are the objectives of gene transfer? Genetic modification of animals may be aimed at improving production of their milk, meat, wool, etc. So the genes have been transferred into animals to obtain a large scale production of the proteins encoded by that particular genes in the milk, urine or blood of such animals. A special case of gene transfer aims at eliminating the symptoms and consequent miseries of genetic diseases also. In this approach, normal and functional copies of the defective gene are introduced into the patient. It is also called as gene therapy. Specific transgenic animal strains or lines are created to fulfill specialized experimental or biomedical needs. So these are all the objectives of gene transfer. There are several approaches that have been used for the introduction of DNA into animal cells or embryos. They are embryonic stem cell mediated gene transfer, microinjection, retrovirus infection, calcium phosphate precipitation, liposuction and electroporation. For the construct of transgenic animals, they usually adopt the first three methods, that is embryonic stem cell mediated gene transfer, microinjection and retrovirus infection. But all these methods will be opted for animal cell handling. Embryonic stem cell mediated gene transfer. This method involves the prior insertion of the desired DNA sequence into an in vitro culture of embryonic stem cells. Okay, the insertion of a desired DNA sequence will be done by homologous recombination into an in vitro culture of embryonic stem cells. Stem cells are undifferentiated cells that have the potential to differentiate into any type of a cell. That may be somatic or and germ cells, and therefore to give rise to a complete organism. These cells are then incorporated into an embryo at the blastocyst stage. Okay, the cells are incorporated into an embryo at the blastocyst stage of development. The result is a chimeric animal. Chimeric animal means it Chimeric animals are the one, they have two or more genetically different genetically distinct cells from different zygotes by sexual reproduction. Embryonic stem cell mediated gene transfer is the method of choice for gene inactivation. This is also called knockout method. This technique is of particular importance for the study of the genetic control of developmental process. It also works particularly well in mice. It has the advantage of allowing precise targeting of defined mutations in the gene via homologous recombination. Microinjection. In this method, DNA solution is injected directly into the nucleus of a cell. A microinjection assembly consists of a low power stereoscopic dissecting microscope in order to weigh the ovum and the entire process, and two micromanipulators, one for a glass micropipette to hold the ovum by partial suction force, and the other for a glass injection needle to introduce the DNA into the male pronucleus. In the picture you can see the assembly. There is a holding pipette to hold the ovum by a partial suction, then microinjection needle with the foreign DNA. It is directly injected into the host cell. The male pronucleus is much larger than the female pronucleus of fertilized mammalian ova. However, in fish ova, the DNA is injected into the egg cytoplasm. Next one is retroviral infection. Recombinant retroviruses produce virions, that they are the infectious agents which are used to infect animal cells and mice embryos. The recombinant retrovirus RNA genome is copied by reverse transcriptase to yield a DNA copy. Okay, the DNA copy will be obtained by reverse transcriptase method from the RNA genome of a recombinant retrovirus. Okay, this is also called as revenue transcription, which becomes integrated into the cell's genome. The DNA copy thus obtained becomes integrated into the cell's genome. The reverse transcriptase is encoded by the retrovirus and is produced immediately after the infection in the organism. The reverse transcription can occur only in those cells which go through S phase of a cell cycle, that is, which must be mitotically active. The DNA copy of the recombinant retrovirus integrates into the cellular genome at random sites. Usually it is not accompanied with deletions or any rearrangements in it. Next one is calcium phosphate precipitation. Here the calcium chloride solution. In this method, the DNA preparation to be used for transfection is first dissolved in a phosphate buffer. You can see in the picture. Then the calcium chloride solution is then added to the DNA solution. The DNA preparation is made by dissolving in a phosphate buffer in order to carry out transfection. Then the calcium chloride solution is added to the DNA solution. This leads to the formation of an insoluble calcium phosphate which co-precipitates with the DNA. Okay. Then the calcium phosphate DNA precipitate which is formed is added to the cells to be transfected. Following, then the precipitate particles are taken in by the cells by phagocytosis. You can see in the picture clearly the DNA calcium precipitate co-precipitate is added to the cells to be transfected. Then the precipitate particles are taken in by the cells by phagocytosis. In a small proportion of the transfected cells, the DNA becomes integrated into the cell genome and producing stable or permanent transfection. So this general approach can be applied to virtually all mammalian cells, and a very large number of cells can be treated easily. But many cell lines do not like the calcium phosphate precipitate adhering to their surfaces or to the substrate. Next is lipofaction, that is lipid mediated transfection. Here the delivery of DNA into cells using liposomes is called lipofaction. Liposomes are small vesicles. They are prepared from a suitable lipid. Here the cationic liposomes have a single lipid bilayer membrane and they bind to the cells efficiently. Probably they fuse with the plasma membrane and thereby deliver the DNA complexed with them into the cells, which brings about transfection. Here the culture and plate cells to the transfection assay taken. Then nucleic acid and transfection reagent mixtures are prepared separately. Then the transfection reagent and nucleic acid preparations are mixed together and incubated. Then the cationic lipid nucleic acid complexes, as I said, forms. The formed complexes can be added to the cell cultures. Then incubate those cells to facilitate the endocytosis of the complexes. The lipid nucleic acid complexes will be engulfed by the cells by endocytosis. Then the lysed cells can be lysed and used for further process. Lipofaction is the method of choice for transfection of mammalian cells in vitro. It has also been used to deliver DNA into live animals by direct injection or intravenous injection. Cationic liposomes have been used in intravenous or intratracheal injection in mice for the expression of marker genes in lungs. The last one is electroporation. Electroporation is based on the principle that high voltage electric pulses can induce cell plasma membranes to fuse. So electroporation is a technique involving electric field mediated membrane permeabilization. Here electric shocks can also induce cellular uptake of exogenous DNA. It is believed to be through the pores formed by electric pulses from the suspending solution. Here the cells are placed in a solution containing DNA and are subjected to electric shocks to cause holes or pores in the membranes. Then the foreign DNA fragments enter through the pores formed into the cytoplasm and then to the nucleus. So here you can see the picture before pulse during the electric current and what happens after the electric pulse. So when we apply electric current, you can see the formation of pores on the membrane of the cells. So electric field induces voltage across the cell membrane causing pores. So through those pores, the genes to be inserted or the foreign DNA fragments to be inserted will be entered through those pores formed. Then after electric current, the membrane gets healed and it is genetically modified though. Thank you.